Good afternoon, everyone, and welcome to Huron University College. I know most of you, but not everybody, so if we haven't yet met, I'm Stephen McClatchy. I'm principal of Huron, and it's my great pleasure to welcome you all here for the 8th Margaret and Douglas Derry Interdisciplinary Lecture. The avowed purpose of this lecture series is to illustrate how the different disciplines connect with each other to reveal an underlying basic unity that has always existed across human knowledge. We're very grateful to Margaret and Douglas Derry for their keen interest in the interdisciplinary work that Huron does so well, and for their idea to make this lecture series a reality. And we're very grateful for their generosity that has made it possible to do this. So thank you so much for your generous support. Tonight, we're honored to welcome Dr. Paul Hebert to Huron University College. Dr. Hebert holds a Canada Research Chair in Molecular Biodiversity at the University of Guelph, where he is also professor in the Department of Integrative Biology. He is a director of the Biodiversity Institute of Ontario and scientific director of the International Barcode of Life project, which is the largest research program ever undertaken in biodiversity science. His early research focused on breeding system evolution and population genetics, but is best known for his work on the rise of DNA barcoding. Dr. Hebert is an officer of the Order of Canada, a fellow of the Royal Society of Canada, and has honorary degrees from the Universities of Windsor and Waterloo. Dr. Hebert has received nearly $100 million in research grants, and his 420 publications have attracted almost 30,000 citations. Among his 91 former graduate students and postdoctoral fellows, 39 of them are now in faculty positions. So an incredibly impressive record, no matter which way you look at it. So tonight, we're looking forward to hearing Dr. Hebert talk about planetary biodiversity, agents of change. Ladies and gentlemen, it gives me great pleasure to introduce Dr. Paul Hebert, Huron University College's 2015 Derry Lecturer.
Well, Stephen, thank you very much for that introduction. That was very kind. It's a great honor to visit Huron University. I must admit, I have thought of Western as a great big, ambiguous university, and didn't have a real sense of these smaller universities embedded in its midst. And I think that's where you really get an education, working in that kind of very personal context. I'm going to talk to you tonight about an issue that's very close to my heart, and I think it's an issue that should be close to humanity's heart, and that's the organisms that we share this planet with. And I'm going to focus on biodiversity, that means, but I'm also going to place it within the context of technological change and the kinds of magical things that we can now do with genetics that may make it better, to make it possible to do a better job of protecting life on our planet. And I want to begin by looking back at my own childhood. I grew up in Ontario, in Kingston, and some of the organisms that meant something to me. And one of them was a cecropia moth, and I'll never forget when I was 6 walking in the backyard and finding a mating pair of cecropia moths. They're the largest moth in North America, a really spectacular organism. We always had a purple martin house in the backyard, and many people in Kingston had them, and we always looked forward to spring and the purple martins coming back and trying to get more purple martins than the people in the house down the street. And Kingston was a beautiful city with elm trees. The streets were just lined with elm trees, and Baltimore orioles breeding in those trees. And we had a cottage on Lake Ontario, and I used to spend many days in Lake Ontario busily wandering around doing damage to the freshwater clams. Not too much damage. I collected them and put them back in, but they were really one of the most conspicuous invertebrates in Lake Ontario. And of course, I had some affection for moths and butterflies, and so monarch butterfly caterpillars and the milkweeds, collecting them, rearing them, was just something special. And at night, when we were at the cottage, we would hear whippoorwills singing their wonderful sound. So those were organisms, 6 organisms that meant a lot to me growing up in Kingston. They're really talismans, I think, of my youth, and no young Kingstonian will feel that, because those species are effectively gone from Kingston today. The situation, those 6 species, isn't an exception, unfortunately. This is the World Wildlife's 2014 report. They've surveyed 3,000 species of vertebrates since 1970, and over that interval, 52% decline in the abundance. And of course, the world didn't begin in 1970, so it's fair to ask what was happening before that, and the answer is a lot of population declines. This is 52% since 1970. It may be a much more dramatic reduction than that, and in fact, if we look at the hotspots for biodiversity on the planet, because we all know that life isn't distributed uniformly across our planet, these are the biodiversity hotspots, the place where there's an intense diversity. They cover 17% of the land surface, but 77% of the vertebrate species on our planet live in those habitats. So these are really important places for life. And if you take a look at the vegetation cover on these habitats, this is what you see. Half of those habitats have now lost 90% of their original vegetation cover. 90%. And in fact, the best preserved habitats, like the one in California, have lost 2/3 of their native vegetation cover. So there's been massive loss of habitat, and associated with that is going to be massive declines in the abundance of species that live in those settings. And of course, this isn't the only problem that organisms confront, the loss of habitat, it's the changing habitat. And our world is warming. Climates are changing, and if you go to California, this is the most intact biodiversity hotspot with about 40% of its vegetation cover in place, but it's undergoing the most pronounced drought in 1,500 years. So if you're living in that hotspot and it gets hot temperature-wise, you may no longer be adapted to it. So the hotspot may be there, but unfortunately you're not going to live. So that's one problem, climate change, and then on top of that is those forest remnants that we're talking about have been fragmented in the most serious way. And in fact, if you look at the forests on our planet, this little graph shows you the distance to the forest edge and the proportion of forests on our planet. And the answer is 70% of the remnant forests on our planet exist within 1 kilometer of a road. The idea of isolation, isolated wilderness in our planet is almost gone. So this is what the other species on our planet confront. And of course, the question is why? Well, and what else is happening? It's not just declines in abundance, it's actual extinctions. You can see this rapid rise in the extinction of vertebrate species. We don't have good information on invertebrates, but there's this very dramatic rise in the extinction of species over the last 100 years. And what's it associated with? Well, of course, it's associated with us. When I was born, there were barely 2 billion people on the planet. It was only the first time humanity crested 1 billion was in 1850. It took nearly till 1950 to reach 2 billion. And now we hit 7 billion in 2012, and we're on our way to 9.4 billion by 2050. This tremendous growth of our species is, we're doing a good job of feeding people. I mean, you know, you hear there are problems, there are regional problems, but if you take a look at grain production on our planet since 1970, we've managed to double it. That's why most people on our planet aren't going to bed hungry. We've been successful in increasing agriculture production in a radical way. And how have we done this? In Canada, we ran out of farming land some time ago. We're not clearing farming land in Canada. There isn't any left to clear. But if you look in the tropical regions of the world, there was land that can be co-opted into agriculture that's being decimated. This is a photograph in Brazil. If you'd look 10 years before, it just would have been green forest. It's now roads, and those roads will grow, and that will be continuous farmland very soon. This is what's happening in Malaysia. Wonderful lowland rainforest sites being converted into oil palm monocultures. Malaysia probably is the most biodiverse part of our planet. So that we can have palm oil, we're doing dramatic damage to habitats that supported an immense diversity of life. It's not just, of course, agriculture is more than clearing land. You need to grow things on that land, and one of the ways that we maximize production, of course, is by adding agents to protect the things that we're interested in growing. So pesticide being sprayed here. It's quite remarkable to me how much pesticide and herbicide and fungicide is used. So these are the figures for the United States. And you can see that it's been stable through time. That's the good news. Agriculture production has increased substantially over this interval, and the usage of these agents hasn't much increased, but when you tally it up, it's 500 million kilograms of active agents per year. There are 300 million people approximately in the States, so that's a kilogram and a half. So look at the person beside you and ask how much, what weight of pesticides, herbicides are going to be required to keep that person alive for their lifetime, and the answer is their body weight. Their body weight in noxious agents added to our environment to sustain our agricultural system, to feed us. It doesn't just kill the things on the fields. It gets off the fields and does damage to other life. One of the other interesting side effects that you mightn't think about with a burgeoning human population, of course, comes globalization of trade and the need to move objects from one side of the planet to the other. And of course, when we're moving cars or we're moving food items, it's often not just those things that move. And so global trade has had an immense effect on biology of our planet by moving things that are pests, that may not have even been a problem in their home nation, but when they get somewhere new, they explode. So humanity is a serious issue, and it's not an easy thing to escape from. Let me look back at my childhood talismans and ask, what happened to those 6 species? And the answer is 3 of them were lost due to invasive taxa. Inadvertent introductions of things that we had no reason or economic gain. They were just transferred inadvertently. And the other 3 have probably been lost because of the chemical dosing of environments. And all of the organisms probably decreased, at least in part, because of habitat loss. So this magnificent moth, the Cecropia, I could walk along the streets when I was a kid in Kingston and see cocoons dangling off of trees. It would be easy to find dozens of them in a day, and now you won't see any. Why is it no longer with us, or why is it so scarce? The gypsy moth was inadvertently introduced into North America in the 19th century and created some havoc in the forests. And so the decision was made to introduce that little gray taconid fly. And that little gray taconid fly didn't do a very good job of killing gypsy moths because they're still here. It actually preferred the larvae of the Cecropia moth, and it's decimated all of the large moths in eastern North America, all of the silk moths and the sphingids, other big objects, things that were bigger than the gypsy moth. So that's what happened to the Cecropia. That was an intentional introduction of a species for biological control that went wrong, seriously wrong. The elm trees that lined the streets in Kingston, why did they go? Well, I think we all know that Dutch elm disease was introduced, and it was introduced into the States in about 1930, and you can see the speed at which it moved across our continent and effectively wiped out this tree. I remember when I was a teenager, all the giant elm trees in our backyard dying because of Dutch elm disease. And of course, we still have elms, but they never get to be mature today. So that was an inadvertent introduction. No one planned to bring in Dutch elm disease. No one set out to do that. What happened to my clams? Well, the clams were introduced as a consequence of shipping in the Great Lakes when there was in fact, my lab was the first one to find a zebra mussel in North America. In 1986, we were sampling. I used to be based at the University of Windsor then, and we were sampling in Lake St. Clair, and a student came in with one of these, and I said, oh my God, I've never seen one of those. Let's take a photograph of it. It may be the last one we see. That definitely wasn't the case. This thing spread like a rocket across North America. And as it spread, it wiped out, it overgrew the unionid clams. We have 300 species of unionid clams. It was a magnificent radiation of organisms with spectacular biology. Their larvae move into fish gills and just all sorts of wonderful aspects of biology, aside from their physical attractiveness. And they're effectively gone. They're all on the endangered species list now, and as one lake is colonized by zebra mussels, within 2 years of them getting into Lake St. Clair, all of the clams in Lake St. Clair were dead. They just overgrew them. And of course, it's not, there's an economic penalty associated with these invasive species, some $500 million a year in damage created by that single species. In Canada alone, in the United States, it's way over $1 billion per year. There's a lot of economic loss associated with invasive species. So those are 3. This is, again, was an inadvertent one associated with ballast water discharge. The birds. What happened to my purple martins and what happened to my nightjares? Well, the answer is they weren't alone. The flying insectivorous birds in North America are under steep decline. The whippoorwill's extinct in eastern Canada now. It's gone from Nova Scotia. I think there were 2 pairs left in New Brunswick. It's going. And this is probably a consequence, no one really knows, of declining insect populations associated with the abundant use of pesticides. One of my colleagues, we had a meeting recently in Guelph, he came from Germany, he said, we're no longer selling window cleaner for car. He had a picture of a German Mercedes sitting there with the car window, and he said, we no longer need to clean our car windows when we go driving in Germany in the summer. There aren't enough insects in the sky. We used to coat them. I don't know if we have the same feeling here, but I can tell you, I think there are fewer insects and many fewer butterflies than I was a kid. And these birds are just running out of food, maybe not necessarily in Ontario, but somewhere en route to their wintering grounds. And so they're in major collapse. The monarch butterflies, this massive overwintering colony in Mexico, 27 acres in 2003, 1 acre in 2013. You have to look really hard. I looked this summer and I didn't find a monarch caterpillar. I did see a few monarchs flying by, but any afternoon when I was a kid, if I'd wanted one in July, I would have found monarch butterfly caterpillars. Yeah, we're doing tough things to life on this planet. And it's not something that's new. I don't have time to talk about all the sad cases. I could talk a little bit about the pasture pigeon, actually, whose last nesting ground was on a farm very close to where my great-grandfather lived, north of Kingston. For all I know, he ate one of the last pasture pigeons I hope that isn't part of my family heritage, but it might be. This is a tree called the eastern sequoia. It was very, very abundant in the eastern half of North America, especially in the United States, but spreading up into southern Ontario. Look at the size of this massive and beautiful tree. You've never seen one like it because it was wiped out. The American chestnut was wiped out when about 1910 a fungus was introduced into the United States and spread and wiped out the chestnut. So this is looking back in time. We may have lost the elm when I was a teenager. This is something that happened when my dad was a teenager or my dad was young. And now, of course, the people that are teenagers today are watching the loss of another tree species, the ash. This is 22 species of trees. The ashes, which we all know here are just being decimated again. This is due to an inadvertent introduction. This little beetle species is going to wipe out all of the ash trees in North America, 8 billion trees. 8 billion trees. They've been valued at $30 per tree, and when you do the math, that's $240 billion worth of damage, that one beetle. $240 billion because we didn't exert proper biological surveillance. We allowed trade to be continued and just externalize these costs, society picks it up. It's no longer controversial. It's been 650,000 centuries, 650,000 centuries since life on our planet experienced its last mass extinction. And that was caused by a tremendous meteorite colliding in the Yucatan Peninsula. It wiped out 20 or 30% of life. This Pulitzer Prize-winning popular novel documents the sixth extinction. There have been 5 prior ones in Earth history. You go to the American Museum, this isn't just the popular press, the American Museum of Natural History. We're in the midst of the 6th mass extinction, this time caused solely by humanity's transformation of the environment. You can read recent scientific papers, Science, 2015, Mark Urban, 1/6 of all species could be extinct by 2100. That's 1 species per day between now and then. 1 species per day. We need to do something about this. History is not going to judge us kindly if we sit back and watch massive numbers of species that share our planet just simply disappear and we do nothing. It's completely unacceptable. How do we do something about it? There have been calls to action. It's been argued we have a moral obligation to protect other species on our planet. It's been argued that we need to protect other species because of their aesthetic value. This hasn't provoked investments. These 2 arguments haven't provoked major investments. We've watched the species disappear. It's been argued that biodiversity is one of the important elements for planetary sustainability. It's one of the 9 axes that we really must look after. And these 3 are all recognized to be in serious issues right now, but there's 6 more that haven't seen as much investigation. Again, it hasn't been enough to provoke major investments, simply saying that we're risking our planet's well-being. I think there may be another way that we can shift societal support in favor of major investments, and that is to recognize that there's something special about humans. We value knowledge. We're questing for it all the time. Humanity spends billions of dollars every year to try and understand its world and everything about it. It's what makes us tick as a species. Stephen Hawking, we spent $700 million as a species this year to fly by Pluto. $700 million. And oh my God, I loved those images. It was wonderful. And I will love every image collected by the wonderful telescopes that are staring out into space. And Stephen Hawking, we explore because we're human and we want to know. Yes, we do. But we should be wanting to know about the things that are closer to us, the things that we live with. The loss of species, every species that's lost, is an irrevocable loss of knowledge. Don't let the dodo fool you. It's these big organisms, when they died, they left bones, and we can mine those bones for DNA, and we can read the genome of the dodo. But most species are far smaller, and when they become extinct, they're a smudge on the forest floor for a few hours, and they're gone. It's not acceptable to just erase this part of our human heritage, the heritage of our planet. Zettel's Traum is the longest book that's ever been written by a human. I don't know how long it took to write, but probably just a few years, maybe 10 years. 7 million characters. The human genome is 7 billion. The book that describes how to make a human is 7 billion base pairs. It's 1,000 times longer. It's an immense book of life. And it's one of many, many books of life on our planet. This one we've read, and we're reading it increasingly. We're fixated on reading the Human Genome book. There are bigger books, the Japanese canopy plant, the marbled lungfish, far larger, 25 times larger than the human genome. So there are bigger books. The average book of life is about 1 billion base pairs. We love knowledge. Even when we've read books, we don't burn them. We put them in place and we look after them. This is the Library of Congress. $600 million a year to look after the books and documents stored in the Library of Congress. That's quite a lot of money. As we look after those books, we're busy burning the books of life all around our planet, every day. I'm determined, and others of my colleagues are determined that we do something about this. That as a first act, we don't simply let the books be burnt. that we do something, that we launch a major exercise to collect, gather the organisms, the diversity of species on our planet, and be absolutely certain that we preserve in each one of these wells the DNA from a particular species. Every species on our planet in a DNA archive. So we can read these books of life at our leisure. It's sort of difficult to read books of life right now. It costs a bit of money. It's not going to cost anything in the future. It's going to be very, very inexpensive. And this is the kind of fallback position. If you're going to burn it, don't burn it, save it so you can read it later. And some of my colleagues say that's a very incomplete way to document life. You're going to have a DNA blueprint, but it's not really going to tell you what that species looked like. It's not going to reveal the majesty that is life. And that's true. We don't know how to take a gene sequence and say, that is what it's going to look like. It's going to have the following features. But in fact, the workflow that goes into gathering organisms, the DNA barcode community that's carrying out this work, we don't throw away the organisms. We collect organisms, and all of these organisms that donate the DNA that goes into those extract wells are kept. They're photographed, they're documented, they're available for humanity to look at. So we'll have the phenotype, what these things look like, we'll know where they came from, and we'll also have the DNA so that we can read the books later. And what are we going to do with the DNA? The Norwegian government sponsored the development of a thing called the Global Seed Bank up on Svalbard, perfect place if you want to keep things cold. And all of the world's important agricultural plants are held in storage in that vault. All the nations of the world have contributed to it. And so the first thing I thought is, well, if they've got it right, let's just ask them to build a second tower, and we're going to put the Global Library of Life into the second tower on Svalbard. And in fact, Norway said, no need to do that. We actually have 3 rooms at Svalbard. One of them's occupied by the plants, another is needed for administrative purposes, and you can have the 3rd room for the Library of Life. So we have somewhere to store the DNA. I suspect we'll actually want to copy this DNA and have a few copies in different places around the planet. It's easy to immortalize DNA today. So let me switch and tell you a little bit about the problem that we confront in doing this. And the problem is simply, we spent the last 250 years describing species on our planet, and we've done 1.7 million species in 250 years. And we're looking at many of the species disappearing this century, and we think there may be 8.7 million species on the planet. How are we going to get the job done before the species have gone? How do you create a library of life if you can't read the book titles? That's the minimum. Well, let's carry on doing what we've been doing. Let's index species through morphology, size, shape, color. That's the way we've been doing it. Does that scale? $250 billion to describe 5 million species. That's what it will cost if we carry on with current trajectories. And that number is a project killer. There are communities that are used to running mega science projects, and a mega science project is $1 billion plus project. And the physicists and the astronomers know that you don't mention numbers bigger than about $10 billion. Large Hadron is $9 billion, Hubble telescope is $10 billion. That's the kind of cash that you can get from the global community for big science projects that are worth doing. So biodiversity science has to get realistic, and we need to figure a way to do what we need to do for on the order of a couple of $1 billion. That's an everyday mega science project. They're launched pretty regularly. Okay. Problem, we don't know how many species are on the planet, we don't know how to tell them apart, what are we going to do? Other sciences have had similar kinds of problems. George Hale wrote to the Rockefeller Foundation in 1928, just before the financial collapse, and said, no method of advancing science is so productive as the development of new and more powerful instruments and methods of research. Instruments, methods. 1928, financial collapse, not a good time to be writing asking for money, but the project was approved shortly after, and Hale didn't get to see it in operation, but it opened and it saw first light in 1949. This is the Mount Palomar Observatory. It was made fantastic contribution to our understanding of our universe. Biodiversity science finds itself in a wonderful position to follow Hale's maxims. We have been presented with new instrumentation. These powerful automated DNA sequencers, which are a technology that are doing absolutely advancing in a magnificent way, and this is part of this answer. Don't tell things apart by appearance, tell them apart by DNA, and use this technology. So we've got a technological advance, and we've also, this little idea, as Stephen mentioned, that bubbled up in Guelph, where we proposed that you don't need to read the whole genome of organism to tell things apart. Just read a tiny little piece of DNA, a DNA barcode, and this will allow you to tell species apart. And so this is the shift in terms of research approach that makes it possible to register all life. Couple the sequencers with this idea that we don't read the whole book, we just read its title. Makes it possible to do great things. And so this is what we're beginning to think about now. Is a $2.5 billion project. It's a mission for planetary biodiversity. And the idea is to ignite this mission as quickly as possible. Got to jump a little bit into science. Hopefully that won't bore you. Tell you a little bit about DNA barcoding and how we plan to do this project. And I sort of mentioned already a DNA barcode. What is it? It's a short piece of DNA. It's a standard piece of DNA. So you read a particular position in the genome. Genomes are big, complicated things, but we want a standard place we could read. And the idea is that it would allow you to tell apart the species in a large block of life. How cool would it be if you could go to the seashore and all of those species, you read just a tiny piece of DNA, and it would immediately tell you the name of that species. It would tell you that this is a cnidarian, a particular type of anemone. This is a type of mollusk. It would tell you the algae. And that's the principle behind DNA barcoding. Find a tiny piece of DNA that's present in all organisms that contains enough sequence variation to tell apart species. So this is an animal cell. That's the nucleus. That's the thing we normally think about. It's massive. In every one of your cells, it's 7 billion base pairs. But if we look carefully at your cells, you'd have these little green fluorescent objects here. These are the mitochondria. And the interesting things about them is they contain a little circular loop of DNA. And you can see all the multiple copies of that. They're all inherited maternally from your mother. They evolve quickly. They have some features. The DNA in that little circle has some features that make it very useful for a barcode. So when we thought about barcoding animals, we selected this little loop of DNA, and we read just the front part of one of the protein coding genes. Just 650 base pairs of DNA will allow you, now think back, 7 billion base pairs makes a human, but we only need to read 600 of them, 650 of them to tell a human from a chimpanzee, a gorilla, or any other animal species on the planet. This is a fly. We've got lots of things in museums today with legs away for DNA. Actually, it's a leg away for DNA. And this is a very hard to pronounce word, but that's the barcode word for that fly. And every individual of that fly species would have basically the same word. And it's very easy with those automated sequencers to read them. And if you're interested in birds, North American birds, and you're interested in seabirds, what you'll find, and here you can see two individuals of a species, and I don't want to dig too deeply into this, but this is a tree that's comparing sequences among members of a species and between species. So here you can see two individuals of the upland sandpiper. They're almost identical. The two members of the long-billed curlew are identical. We've just done a single whimbrel. Two individuals here of the short-billed dowitcher, identical. And you can see the deep branches, and this is the sequence divergence between species, between the short-billed and the long-billed dowitcher, which I'm sure bird people can tell apart, but I'm quite certain I couldn't, especially if they weren't in breeding plumage. Although maybe I could. Short versus long bill. That sounds like something you could work with. North American birds. Reading those little barcode region, that little region, the front end of the gene, allows you to tell all those bird species apart. No difficulty. You could go to the tropics. When we first proposed this idea, people said, you live in Canada, there aren't any species in Canada. It's very easy to tell them apart. You've only got 3, polar bear, beaver. Anyway, this is Costa Rica, where there's lots of silk moths, and the important point is you're reading the same piece of DNA, and we can tell apart all the saturnates. And Dan Jansen, who spent his life working in the rainforest, was like, oh my God, this is wonderful. This is one of the better days of my life when we presented these results to Dan. So we did this for about 4 or 5 years after we proposed the idea. A lot of people were skeptical. but we managed to gather up some resources and gather a lot of data. So that in 2007 led to a meeting in Guelph in which we proposed that we would build an international alliance to create this International Barcode of Life project. And we thought, imagine a world in which you can identify basically any organism on the spot, anywhere on our planet in an instant. And that's the world that the barcode community is trying to bring to humanity. This possibility of just touch a thing and find out what it is. Go for a walk in the woods with your child or grandchild and touch the organism and find out everything that humanity knows about it. And we said in 5 years, we want to barcode 500,000 species on our planet. And by the way, it will cost $100 million. And no one wrote a check for a few years. But by 2010, enough people had written checks, and I'm proud to say that Canada made a very substantial contribution to this project, as did many of the other nations around the world. And we had a scientific meeting in September 2010, and we launched the project in Toronto and in Nagoya. So that was the beginning of the Barcode of Life project. And this is our planet in 2003. If you look really hard, you're going to see a little dot here in Guelph. That's how many barcode records we had in 2003. Not many. 2005, starting to see a bit more action in different places. 2007, reds are good, yellows not as good. 2009, 2011, 2013, 2015. So our dream is to paint the world purple. Everywhere. Thousands of specimens, every site on our planet. Understand the diversity and distribution of life. We set our milestone to barcode 500,000 species, and guess what? In August of this year, we reached the 500,000 species. It's never good to say you're going to do something and fail, and we did it. So this was quite a nice day for us. However, we haven't done all the species on the planet. We've done 500,000, and the problem is we weren't quite certain how many species there are on our planet. Maybe 10, 20 million, because now we're getting ambitious. We want to do everything. And if you ask people for money, they'll say, well, how big a job is it? And we say, don't know. That's not good. So we've used Canada as a role model. Our nation is 7% of the global land surface and 1% of all biodiversity. And we think we have about 10,000 species of plants, 85,000 species of animals, 15,000 species of fungi, and 10,000 species of protists. About 120,000 species known from our nation. We went into the herbaria in our nations, the places that keep plants dried on paper, and found we're very easily able to liberate DNA barcodes from them. And so we finished the vascular plants of Canada. So the plants of Canada are going to get finished in short order, and I don't see any problem doing the plants of the world. The largest compartment of animal diversity in Canada are insects. And so we decided we'd have to do insects, and unfortunately, insects in museums aren't ideal to work with. We did know there were about 30,000 species known, but because insects in museums weren't easy to work with, we realized we had to go out into nature and try and recollect all these species. And the question was that going to be possible or not? We have recruited all sorts of help. And especially low-paid help, like school kids. Turned out to be absolutely perfect. They absolutely love this project. This spring we had 68 schools across Canada deploying these malaise traps in their schoolyard, and it's a fantastic project. You know, this isn't citizen science, this is school kid science, and the data doesn't end up on the cutting room floor of science. It gets into the Barcode of Life data system. So they collected for 3 weeks. And those are where the schools were located across Canada, and we've been doing this for a few years. So we knew it was effective, and the kids blog about it, the kids tweet about it. It's pretty cool. And those school kids collected 2,929 species, 10% of the insects known from our nation. So that's one way we've got them. Then you can find dumb faculty members who are like nothing better than to have a malaise trap in their backyard. That's my backyard, that's my malaise trap, and I love going out every week and taking, I'm not panning for gold, I'm panning for bugs. My grandson is absolutely wrapped in the things that come out of this thing. So How many, so there's Guelph, there's my backyard, trap, bottles, barcode. 4,700 species of insects that I share my backyard with. 16% of all the insect fauna known. I never imagined I would have that many species of insects in my backyard. And then we went to Canada's national parks, and we set a Malaise trap up in every one of Canada's national parks with the help of Park Canada. And there they are. Over just 3 years, we managed to sample them with the help of Parks Canada staff. 29,000 species known. We collected 39,000 species in the parks. That's how many were known from Canada before we began. There's where it was when we began. We're now at 57,000 species of insects in Canada we haven't tapped out. There's a lot of insect species in our country. 191% of known insects, and 49,000 species of animals were known. We're now at 70,000 and rising. So we're finding through this approach lots of new species. And I admit they're not new bear species, or they tend to be small and rather gray and boring. Can we barcode the world? I'm convinced we can barcode the world. We barcoded Canada, almost. It's going to take another few years to progress things, but let's look ahead. This is what we've done so far in Canada. We've done seaweeds, all the vascular plants, all the vertebrates, many of the insects, got into land and fresh water. We didn't get into some places yet. We've done very little in marine environments, some small invert groups we haven't done, and so there's work to be done. We've collected 1.5 million sequences in Canada so far. I think we're going to need 1.5 million plus to finish the job. But, you know, that's, if you've done 1.5 million in 5 years, 5 years now we'll be done. How cool will that be? Take a look at our planet. Yeah, it's a little bit bigger than Canada, but we're only talking about 300 million records that need to be analyzed to register all life on our planet. Remember, this is going to give us this reference library, the Library of Life, during the course of this. However, you're not going to do 300 million specimens without a significant investment. And that's where the Planetary Biodiversity mission comes in. We're probing species with DNA. The Human Genome Project is the only other mega science project that's ever happened in biodiversity science, and we're determined that the second one will be the Planetary Biodiversity Project. What are we going to need to do? We're going to have to recruit more involvement than we had in the last round in the International Barcode of Life project. We had 25 nations. We're looking for 50 nations this time to be involved. Many of them will be archival nodes. They'll be collecting the specimens. You can't collect elephants in Canada. Well, you can at a zoo, but you need to go to the habitat where species, and so we need to have a lot of participation around the planet collecting organisms. And then we need a few biotron facilities, large analytical facilities like the one we have in Guelph that's going to analyze all this, process it, save the DNA, send it up to Svalbard, replicate it and put it into repositories around our planet. Analyze 20 million specimens each. That's basically, we can do that with our core facility over the next 20 years. So here's the plan, 2016 to 2020. That's where we clarify our scientific goals. That's where we build momentum and secure the funding. 2020 to 2040 is the time frame to carry out the work. What will we get from this? We will get an inventory of species for our planet. We're going to learn a lot about the ecology, evolution, and morphology of them along the way. They'll all be 3D imaged. They'll all be available, living on the web. The Library of Life, we're going to have this resource for genomic analysis that will be held in perpetuity for humanity to read at its leisure. And the other thing that we're going to do is, this work is going to create the possibility for advanced biosurveillance. When you think about what I've been talking about so far, building a registry of the species on our planet, that's like saying, I'm going to create a thermometer and I'm going to know if it runs from 0 to 100 Celsius. We're just calibrating. We need to use the thermometer. We need to read life on our planet, and so that's why this new technology of next-gen sequencing is going to make it possible. Those bugs in the bottles in my backyard, we're going to be able to read those in a flash on one of the new sequencers. We're going to be reading life like we read temperatures or rainfall today. Imagine what we'd be doing in terms of global warming if we weren't reading thermometers. We haven't been reading life. We need to read it. What are we going to get from this? I hate getting back to this basics of, gee whiz, you need to invest in our project because it's going to do something economically good. Because physicists and astronomers don't talk that way. It's grand idealism. But the fact is, this is a project with paybacks. And that's why it should be so easy to raise the funds for this project. Imagine if we ringed the ports of Canada with monitoring stations so any species that wasn't native was recognized immediately upon its arrival, and we would take deterrent action. The zebra mussel, the emerald ash borer, if we stop a single species for 1 year, it would pay the entire project back in one nation. I mean, it's a complete no-brainer. So halting invasive species is one where you can put an immediate, tangible economic benefit associated with the work. Having that DNA, we're starting to become close to doing magical things with organisms. Of course, we know genetically modified organisms are moving into agriculture, but there's a much broader possibility of using genetic modification. This is the chestnut. This is the emerald ash, and this is my cecropia. I want them back. The chestnut was wiped out because of a fungus that released a single substance, oxalic acid, in very high levels into the chestnut, and that killed the tree. Something very neat has just been done, and that is, well, we knew the problem, so take the oxalate oxidase gene from wheat and put it into the genome of the chestnut. And guess what? We now have created a genetically modified, not we, people at Cornell have created a genetically modified American chestnut which is completely resistant to this fungus. We will be growing American chestnuts again. And with global warming, Canada will be filled with them right up to Resolute. Genetic modify ash trees so they're resistant to the emerald ash borer. Trees are resistant to, the trees that live with the emerald ash borer are resistant to it. We need to find the genes involved in that resistance and move it into all of our 22 ash species. Insects are resistant, not all are susceptible to that taconid. Find out the genes responsible for resistance, move it into the cecropia and all the silk moths so the kids of the future can look again at the large silk moths. Watch out. This is one potential problem. We could go too far. We could embed all of our tree species with genetic protection that kept them from being damaged by any insect. If we don't tailor this genetic modification carefully, we could end up with green deserts. We could end up with magnificent forests that nothing could eat. There will be no insects, there will be no birds, these will be empty forests. So this has to be done with intelligence. It's not simply a matter of genetically modifying in the simplest possible way. Resurrecting biodiversity. There's been a lot of talk recently about, and how wonderful would our world would be if we had dodos running around. I just think they're wonderful animals, but why not mastodons? We're now able to recover genome sequences from these organisms, and it's absolutely a possibility in the future we will be resurrecting the species that we decide we want to resurrect. Where we have the DNA, and with these big things, we can do this. With the small things, we don't have the DNA unless you invest in the library of life. And then we can bring back potentially every species. When humans get lonely on this planet, when they wake up someday and say, I miss hearing bees, I miss not seeing any butterflies. My grandfather told me there were once a thing called butterflies, the Soylent Green sort of moment when humanity finally realizes we need to reverse this. These DNA extracts are going to make it possible to resurrect life. How are we going to get this money? I'm determined that Canada play an important role in this, and the very first thing I think I'm going to do is ask the Canadian Mint to start paying royalties on the use of biological images. It's never licensed. It's never asked a maple tree or a beaver or a buffalo for permission to use their images. And if we just get a 1% royalty, it will create $28 million per year, which will be more than enough for Canada to support its commitment to the Planetary Biodiversity mission. Okay, that's sort of tongue-in-cheek. The fact is, Canada is a very rich nation, and we invest in projects. Just last fall, the Canadian Astronomical Society called for a $300 million contribution to join with other people in building the 30-meter telescope in Hawaii. And at that point in time, I'm not sure that we had a science-friendly government, but the fact is that Canada invested $250 million into that project, which is now undergoing construction. We have money. The Canadian Stem Cell Foundation called for a $500 million investment in stem cell research, and it hasn't all been given, but one project at the University of Toronto received $100 million to advance that work. So Canada's a rich nation. We can definitely afford to participate in this project. And there's our little role there. These are the three mega projects that were just launched this year. $2.5 billion, $1.5 billion, Giant Magellan, $1 billion, and there's Canada's share, the $250 million. 3 telescopes, the goal to see more sharply the edges of the universe. Love it, love the images. Think we should be focused at business closer to home. Okay, final slide. We're living in an exceptional moment in Earth history when humanity, the rise of humanity, is placing at risk many of the species on our planet. We have an opportunity, because of the advance in technology and concept, to do something about it, to not just sit back and wait. The community, the biodiversity science community, has shown that it can deliver. It ran a $100 million project out of Canada, and we're talking about a project that may be 25 times larger. In contrast to every other mega science project that Canada has participated in, where we've been also rans, this would be a project that we could run, that Canada would be the leader in, that it could take pride in playing a central role in advancing, protecting biodiversity on our planet. Carrying out this project is going to, associated with it, are these prospects for genetic modification in the future. It's not all gloom and doom. It's a possibility of rescuing species at risk. It's a possibility if things go terribly bad in certain parts of the world that we could bring these species back in the future if humanity decides that it's important to them. My work, our work at BIO, has involved 243 people over the past 10 years since we began this effort, and these are their names in Wordle form. I hate this slide because it's very cold. These people are very passionate about what they do and have made this possible. They've got tremendous world-class expertise. I'm also incredibly proud and happy to live in a country like Canada that's provided the support needed for our work to advance. Organizations like these have played a key role. We couldn't have done it without their support. Stephen mentioned the substantial investments that have gone into building infrastructure and allowing Canada to lead this project. If you had been in Toronto in 2010 in September, you would have seen one of our happier moments when we launched this International Barcode of Life project and managed to convince the tower to run a DNA barcode up it. And the barcode was that of Castor canadensis, the Canadian beaver that builds the largest structures on our planet, one log at a time. And we're advancing knowledge of biodiversity on our planet, one barcode at a time. And thank you very much for listening. Douglas and Margaret, thank you very much for sponsoring this seminar. It's a great privilege to be associated with you. Thank you.
We do have time for questions if you'd be willing to entertain a couple.
As long as they're easy.
Questions, Colin?
Yes.
I noticed on some of your slides with the trees and the moth, there's a little TM on after the description.
Yeah.
Trademark?
Well, that was a joke.
because there's a big issue about once you change that, people, corporations are gonna wanna own things. It'd be a big mess.
Well, that was, in a sense, it's a warning. And I think it's absolutely possible that some of this work will be led by private sector. I think it's not inappropriate, actually, if people want trees growing in their backyard, if they want to have a maple tree 20 years from now, actually, I would say the maple trees are the next tree at risk. I'm quite expecting we're going to lose sugar maples. They're in damage right now. So I'm perfectly happy to see the private sector move in and do things that society needs being done. That's the way a lot of work on our planet gets done. Yes?
If you can identify DNA particles of extinct species and hopefully bring them back, how would you go about prioritizing which ones you would bring back?
Yeah, well, that's a very good question. The first thing is there aren't that many species where we will have potential to bring them back. It's a handful of very large organisms that died relatively recently under very favorable conditions for preservation. I wouldn't be surprised if we're talking on the order of no more than 100 species. If we rely upon current technology and what we know about DNA, it's too fragmented. There isn't enough of it, except in these special circumstances where we got bones, where we got mastodons frozen in the tundra. These are the sort of poster children for this approach. So I think it's going to be a handful of species that will decide. Pasture pigeon, could we bring back Martha? Perhaps we could bring back Martha. Would Martha live as a solitary pigeon when it was an intensely colonial bird? I'm not sure. We might have to bring back a mega flock of these things can be done. I would absolutely love to bring back some of the more charismatic organisms. I mean, they deserve to be here. It's humans that wiped them out, right? We ate them out, the moas. I would love to share my planet with these things that we drove to extinction within the last 10,000 years, and there were some pretty impressive organisms. I'm also really worried about many of the large organisms that we share our planet with today. They could be gone very soon. Their populations are in collapse. We have what people refer to as an extinction debt. The populations are declining. They had a bank balance, if you will, 10,000 specimens, and they're on their way down. They're in negative growth, just like bank accounts. If you're not getting positive interest, you're going to be zero balance before long. And that's what's a lot of species on our planet. They look like they're still here. They're the living dead. They're species that are going to disappear. So those are the things that we want to bring back. But ultimately, we need the environment for the organism to live in, right? You simply can't, we're not going to keep the pasture pigeons. I wouldn't find it very satisfying to keep them in an aviary. I would find that not where we're trying to make this go. I have a hope that humanity in the future becomes much more intelligent about looking after the planet and satisfying human needs, our need for food and shelter in a much less frightening fashion than we currently employ. Renewable energies and all the solutions that people have proposed. So I'm not suggesting that we truncate humans, but I think that's something that we do need to think about pretty seriously. We can't allow human populations to just continue. I mean, there must be a limit to growth on our species. So it's a very complex ethical issue. I'm no ethicist. And I think scientists only see good in what they do, and the rest of the population sits back. That's a terrible idea. And I think this will be one that will be inspected in that way.
But it's sort of an inverse ratio, isn't it? Human populations are going up and environmental populations are going down.
Yeah, well, when people look at how many Earths we need to sustain current human population, it's about 1.5 if you tried to do it sustainably. So we have to shrink human populations, or we have to figure ways to carry out our lives in a much more prudent fashion than has so far been the case. And we're starting to see signs of humanity waking up. I just hope it's not too late for most of the organisms on our planet. And that's why this is really, when we held a conference in August, and we had 600 people from 60 nations, and there were a couple of talks given by people in Southeast Asia, and when they gave their talk, my feeling was I should run out of the conference and go to Southeast Asia and start collecting organisms, because it would be the only chance that we would ever have to preserve the DNA of these species. What's happening in Canada is nothing. I mean, you know, life is relatively, the organisms in Canada are relatively secure in comparison to what's happening in many of the biodiversity hotspots, which are under intense human pressure, very impoverished nations. This is where life is really at risk. I think we in advanced nations have some responsibility to try and caretake the planet. Some special responsibility. And the amount of money we're talking about here to do this is just so minute. This is a chance for Canada to really rescue its reputation as being a custodian of our planet. Yes?
Thank you very much for this presentation. Certainly your enthusiasm and commitment to this project comes through. And my question comes out of the answer that you just gave, in that, can you speak to the politics of this project? I appreciate your trying to shame us into this. I appreciate your strategy. This is about knowledge gain, which is speaking to the right audience for that. But how realistic is it for you to run away to Southeast Asia? Can you speak to the politics of trying to launch such a global project when there is inequity?
Well, there are inequities, and one way to ensure those inequities are extended is by wiping out life on the planet, because the places that have most to lose are those very places that you're talking about, the developing world. They're the ones that have the potential to benefit from a bioliterate society and from all of the things that will come from the discovery of life. So we've actually found a remarkable uptake and willingness to participate in this project. This is not a project run by Northerners. This is a project that involves deep involvement, and when you think about what happened with cell phones and leapfrog type technology, they didn't lay landlines. In much of Africa, people use cell phones. The developing world, with its immense biodiversity, is never going to develop the taxonomic infrastructure, these very expensive buildings that rely upon very low productivity in terms of species. They're never going to register the biodiversity that way. They see DNA barcoding as the chance for them to inventory their biodiversity and protect it. So we have a lot of involvement from nations in the developing world. The First Nations, after Canada, to set up barcode networks, national barcode networks, were in Mexico, Argentina, South Africa. So that's a hopeful sign, I think, the participation. And I'm determined that we carry this on and extend it in this expanded project. There is a difficulty of resources. And I do feel that rich nations like ours have a special responsibility to pony up the $250 million. If you saw the investments in telescopes, single universities ponying up $200 million to help build a telescope in Hawaii. There's that kind of money in all sorts of places. In rich northern nations, $2.5 billion to carry out this project, it's just a trivial amount of money for what we're going to provide. And the rich nations have to do this for the developing world. The other thing, this is a grand opportunity for the repatriation of knowledge. The large museums of our world, like the Natural History Museum in London and the Smithsonian, have been a large sucking sound on biodiversity around our planet. They've been going out on expeditions and collecting organisms with no permits and basically bringing them back to these central repositories. And if someone in South Africa wants to study the species, and they have to make a trip to Washington or London. Through barcoding, we can repatriate this knowledge so people in the southern nations will be able to access. And I think this is very much in keeping with museum curatorial traditions and legislative actions. The other thing I didn't have time to talk about is that this work is seeing a lot of pickup in policy framework by organizations with a global mandate, like the Global Biodiversity Information Facility and the Secretariat for the Convention on Biological Diversity. So they're actually providing money through Japan, the Japan Biodiversity Fund, so we can train people at our institute. We have 4 there now. We had 10 this summer from places like Bhutan and Nepal. People in the developing world that hold positions of influence in the biodiversity portfolio. So there's a long way to go to explore policy implications And that's why we need people like you, if you have expertise in this area. You know, this isn't a biodiversity project anymore. It's changing, it's morphing into something that's got a lot of dimensionality to it. And it's going to be a strong biodiversity core, but it's not going to be a biodiversity project. It can't succeed. Yes?
A couple of questions. There's one there and one here.
Thank you very much for your presentation. There's a lot to unpack here. But just in your answer to that last question, one of the things as you were talking that's really struck me is you don't talk very much about oceans. And it strikes me that in the answer to the previous question, you talk a lot about nation states, and aquatic spaces don't necessarily fit within that framework. And so I'm wondering if you could talk a little bit about the implications of this project in terms of aquatic biodiversity and how this project plans to be.
Well, that's a great question. And I must admit, I live on land, and so do most humans. So that was my sort of, you know, I mean, you don't want to blow the budget beyond, you know, we're trying to do sort of, this is a group of butterfly collectors, basically, right? People that ran around with a butterfly net and give me $5 and I can carry out my research program. No, we need $2.5 billion. So there's a bit of a sticker shock. I mean, when astronomers come to your particle physics, come and say, I need $10 billion. Oh, you only need $10 billion this time? So we can't, I was very conscious about just moving the budget into the point of unreality and people would run for cover. So I said, where are we doing most damage? And the answer is, I think on land and certainly in freshwater. It's absolutely being decimated. So the idea, talk about land, but freshwater is part of land embedded within it. But when we had this conference in Guelph, there were a number of very senior people at this who got very interested in this idea and came and whacked me over my marine ambivalence. My view is, yeah, the big fish are in trouble. And they said, nope, you're pretty naive. And I said, yeah, okay, tell me more. Yes, huge problems in our oceans. The oceans are being acidified. And, you know, this is a systemic problem. On land, we've got local problems. In the oceans, we have the potential of acidifying the oceans, so many, many species being lost. We have to do the oceans. The happy news is, because oceans don't have many barriers, we believe, based on current understanding, there aren't many species in the oceans. So it's like the decimal point. We might only need another $500 million to assemble information on oceanic life. And we plan to do that. My rewrite of the white paper on the Planetary Biodiversity mission included a much stronger focus on marine for this reason, the fact that it's not just on land we're doing damage. Did I answer your question? I thought there was a second part of it that was.
Well, I mean, there's also the policy implication, I think, around, in the way you responded to the previous question, a lot hinges on the nation state, and the oceanic spaces often fall beyond them.
Yes, and that's interesting. Of course, and so does Antarctica. And mind you, there's only 3 species in Antarctica. No, there's not much. We could do Antarctica in an afternoon, perhaps. No, these are really important questions, and that's why we need advice and participation from individuals that have done deep thinking. I couldn't say anything except superficial things to you.
Thank you for coming and speaking to us, and thank you, Mr. Derry, for organizing it. I'm not a scientist. I have a business background, and the thing that strikes me about this is we've got to get the word out somehow. I mean, and we can't tackle the whole world as much as we have to eventually. We've got to tackle Canada because that's kind of within our realm, I suppose, of the possibility. We've already made so much great progress. But I've never heard of this before. Like, I'm surprised that I haven't been more aware of this. And it's wonderful work. Somehow we got to get the word out. To me, it's a no-brainer between somebody studying the stars and this is right in our backyard. The stars, I mean, they're beautiful to look at, and it'd be nice, but we could use that money for something a lot more worthwhile. And I think we somehow have, I guess I'm making a statement more than asking a question there. But how can you get the message out more?
Well, I think you've made a very important statement, and I mean, I think it's an indictment of the science that I am representing tonight. The biodiversity science community, it's not a complete indictment in that we lack the technology. I mean, in 1993, we started to get automated sequencers, and they were targeted towards humanity. And then we solved a couple of problems that made it possible to look at life at large. So even the prospect of thinking the way we're thinking now, it was impossible. I mean, and in 2003, when we wrote this first paper on DNA barcoding, the venom we got from members of our own community that said, this is absolutely no way this can possibly ever work. And a foundation, the Moore Foundation in California, actually gave me about $4 million to, they thought, to watch it fail, crash and burn, but at least it would, we'd have exorcised this demon that this thing wasn't going to work. So that's only a decade. And before that, biodiversity scientists, if you worked on fish, you talked to fish people. Just these very narrow silos, because that's the only common language you had. It's got scales, it swims, it lives in the oceans. You had something to talk about. But now with DNA, we have people at these conferences, we're all talking the same language. So it's very new, and the culture, and you look back to Hale, when he needed his big telescope. I mean, if you look back 50 or 70 years before that to Darwin, and Darwin's analog, Herschel, they built their own telescopes. Darwin paid £10 for his microscope. He didn't quite build his own microscope, but it was almost on a parallel. And then astronomy moved into this, we can build big mirrors. We can look at the universe. And they had to join forces. So they developed this culture of collaboration and cooperation. They've got a well-oiled machine that publicizes, and we've got tiny voices. And so it's a problem of our community, but I share your belief that this biophilia that lives inside all of us is a compelling advantage that we have when we go and speak to people about doing a big project like this. If we get organized, we will be able to drive this forward, but it needs help and advice. Thank you. I would say it all is. I would say it all is in just the sense we mentioned. I mean, every one of those DNA extracts, every one of these specimens that we have analyzed at Guelph is stored at -80 and may be used at some point in time in the future to rescue species. We're working with the biggest conservation programs on the planet. Daniel Jansen is, I mean, there are only a handful of people that have won 2 Nobel Prizes. There is no Nobel Prize in biodiversity science, but Daniel Jansen, who leads a massive conservation program in Costa Rica, has won the equivalent of Nobel Prizes in biodiversity science, the Blue Planet Prize, the Kyoto Prize, the Japan Prize, and so on. He is completely embedded in this project and feels that no conservation project on our planet can possibly proceed in the absence of DNA barcoding. And we're seeing this in place after place because you have to, if you want to conserve things, you have to know where they are. You have to know how many different things there are, right? So how do you develop a conservation program? You can develop conservation programs for bird species because we've inventoried them. We do a pretty good job of telling them apart. But after birds and mammals, and that's the fixation, right? All the big stuff. But all of the other stuff is neglected. And what we're finding out in these early days of censusing things, we thought the little stuff might do better than the big stuff, but it turns out we may be losing it more quickly than big taxa. That's one of the startling early conclusions of this work. Next week, I'm meeting with the chief scientist of Parks Canada, who's looking at how he might embed this work into a long-term surveillance program in Canada's national parks so we actually know what's happening. Our school programs, this is conservation in action in that school kids are the people that are going to conserve nature. I want to expose every kid to this concept of biological diversity because most of them are sitting playing on computer games, unless you get them out in their schoolyard. And they're making discoveries. And there's something quite special about that. When you, as a child, I found the first representative of this species in North America. And that's happening in schools right now in Ontario. So there's a lot of dimensionality to this, and I think we can't focus too narrowly on the narrow concept of conservation, if that means to you just habitat preservation for some selected. Stuart Pym and I have serious arguments about this. He's all for, I need to build a little bit of connection between these two patches of forest to save this monkey species. I'm not interested in that. I'm interested in the planet.
Thank you very much. This has been an absolutely fascinating, wide-ranging talk. It started a little depressingly, but what an inspiring project. And you're certainly a wonderful spokesperson for this work. We didn't see the scientific depth that you bring to this. I fear we'll be lost in most of the audience, but we certainly see the passion and your ability to communicate this to a wide audience. We're so grateful for you for being here. Thank you so much.
It's my honor, Stephen.